Yes. Yeah. Yeah. Hi. One person has joined. Yeah. 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 Because there is still. Yeah. Yeah. Three now. Three. Yeah. They're coming. Okay. Yeah. Sure. Yeah. 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 Bye. Uh, Hi, can you all hear me?
Yes.
Okay. Should we wait for another 5 more minutes? Maybe others will join as well. Yeah, we'll wait for 2 more minutes. Yes, because yeah. Hello, Viba, or anybody from learn to upgrade site is
ट्राई
here?
करके 2 बार फेल्ड बता दिया है।
Huh?
पता नहीं बैंक कनेक्ट नहीं कर पा रही है ये। वही पब्लिश में तेरे को बोलेगी ऊपर से लेके आओ क्या 70 रुपए 95 रुपीज ट्राई करेंगे नंबर पे हो जाएगा ना भैया नंबर पे ट्राई कर हाँ एटीएम छोड़ के
Hi, Viba. Hello. Hello, everyone. So I think we will start because it's 3:05 now. Okay, I hope all of you can hear me clearly. Can you? Yeah.
Yes, sir, we
Okay.
can.
Okay. Yeah. So I'll go ahead and I'll start now. Um, uh, yeah. So there are before we go into some of the various methods that are used in data analysis. Yeah, and also it's so let's go to the very basics. I mean, there are these semantic terms, data analysis and data science. Okay, or today these are some of the topics we will just scan through or quickly cover. The first one will be the difference between data analytics and data science. Both are interchangeably used in some instances, but in general, data science is a little more kind of a because it is on macro level and it also involves research, as you know, science, the word itself tells, and analytics is mostly using existing methods or softwares or tools to analyze and get some trends out of your data. Yeah, and also in scientific research, why is data analysis very important, especially in the present scenario? As you know, in life sciences fields, there are these large data sets coming from genomics, as well as many other sectors. I mean, including healthcare, there are lots of data, both electronic health records, imaging, and yeah, you know, the entire range. And also in data analytics, what are the various methods that are used? There are multiple statistical approaches that are used. Again, they depend on your data, the type of data. And again, anybody who has done some data analysis, you will realize that even images can be converted into numbers, as you all know, right? Because the intensity and uh, even the colors can be coded using the RBG. Yeah, and among the various statistical tests that are used in data analytics, there is a specific branch called AI and ML. ML stands for machine learning. AI is artificial intelligence, and these have the most potential in terms of because almost every app that you see, or yeah, everything from Google Maps to Amazon, everybody relies on these machine learning models. These AI and machine learning models, and also to understand their scope. I mean, at the moment where AI and ML stand, I mean, what kind of tasks can they do? So we need to understand, because machine learning is built on human learning only. It was trying to mimic a human brain. As you all know, machines still cannot replicate a human brain. I mean, in some scenarios, in some other scenarios, machine learning is much better than human learning. And yeah, and also there are some certain types of AI and ML. Sorry. Yeah. Uh, and we'll also quickly go through some of the success stories of AI and ML, because that will give you a context, because many very good apps, and there are people have started really making money out of these models. Yeah, and finally we'll end with how it is used in drug discovery. I think some of you already know there's a field called CAD, where computational methods which use many of these data analytical tools or algorithms like machine learning methods and all, they have been very successfully used for discovering new drugs or analyzing the drug discovery data. And also, as I told you before, in genomics is where data analytics has the greatest potential, because the data is so large. I mean, sometimes it is several hundred gigabytes, or sometimes it's terabytes, and that's just for one person. I mean, one so you can imagine it's almost impossible for any human, or even if you ask 1,000 people to sit and analyze without computers, they cannot do it. I mean, it's not possible. So we need uh, uh, yeah proper data analytics tools. Yeah. And so let's start with the first slide. So the first, as we discussed, so the difference, these are mostly semantic terms, but there are certain differences. I mean, the first major difference is, as the word implies, data science also involves developing new methods. It's not just applying. I mean, if you can see, uh, yeah, the skill sets required, right? I mean, both require programming, but here you have to develop. So you, because using advanced statistical knowledge, you have to develop new methods to analyze your data. Um, yeah, and also data science professionals generally work on much larger data sets because those are much more challenging, and they heavily rely on machine learning and artificial intelligence and this word called big data, the large data. Yeah, and also the data is mostly unstructured. So unstructured means, so structured data, something like your Excel table or your, yeah, some of these delimited files, they can be considered as structured data. But suppose you're asked to extract data from a website or from a text file, it's much more difficult. I mean, people still can do it. XML, HTML files, people can still extract data using these, they're called parsing tools. Yeah, so you can do that, but it's a much more complex kind of a, yeah, analysis. I mean, you often use unstructured data, but in data analytics, because data analytics people are generally, they don't do much of an innovation. They just use what is already there. So even the data is structured, it is in some kind of a table format. And so just like, I mean, suppose you're given a table, you will open it in Excel and do various transformations. So similarly, data analytics person also can do. Yeah. And so data analytics person's main role is to uncover certain trends. Yeah. And in terms of various tools, I mean, here you can see, because nowadays no one statistical software can really keep up with the advances that are happening. So mostly people use open source only, like Python, R, and SQL is used for extracting data. It's a, as you know, right, database tool. Uh, yeah, it's a database query tool. And people also use Java, but to a limited extent. I mean, there are things that you can't do with Java, but Python and R have become the go-to uh, yeah, programming languages. And yeah, and again the big difference, as you can see, when in a data analytics person skill set, you won't see machine learning, software development, these kind of a thing. He generally applies existing tools including BI, yeah, and Microsoft Power BI, but they have to be good at giving proper visualizations for there. Yeah, because when you show that certain trends, like suppose you want to say that uh, let's say COVID severity is correlating with, let's say, BMI. Okay, you can just give a number. Okay, it looks like there is a very strong correlation, or you can give a specific number. There is the correlation coefficient is 0.8. Instead of that, you can also show it like a plot. There are these scatter plots. So there are many other visualization tools which a data, yeah, which a data analyst should be aware of. Yeah, and data science, because they're dealing with large scale data, they are not really concerned about presentation or visualization, but they're very good at analyzing the data, yeah, developing new algorithms and softwares. Okay. And so the next, yeah, so from now on, I'll make it big. Yeah. And the next major aspect is, I mean, because you're all pharmacy and biotechnologists, so you would ask, why do we even need data analysis in these fields? And because the whole idea is to understand, I mean, understand or make certain predictions about, let's say, humans, or for example, if given a drug, can you predict will he respond or not? Or given a person's current health profile, let's say you do a health screening for a person, so there are many vital stats, right, like BP and weight and all these things. So suppose given a person's health record right now, can you predict in 10 years time what kind of a diseases would he be developing? I mean, so yeah, for that you cannot just have only one time record. You should have a series of records, let's say now, and a record 6 months before, a record and 1 year before, like that. I mean, if you have a series of records, so again, using mathematical models, you can make the prediction, okay, in 10 years time he will develop diabetes or cancer or whatever. So those are possible, yeah. But in I think this kind of a figure most of you are aware of. You can the whole idea of this is that you can reduce everything into mathematics, right? Like any of you who are who know chemistry, right, would know that most of biology can be explained with chemistry. And as you know, biology is nothing but various molecules, or yeah, or even tissues. I mean, as you know, right, see, our body contains multiple organs or tissues, and each yeah, and each uh, and each organ or tissue is composed of multiple cells, and each cell contains multiple proteins, DNA, RNA, uh, some certain small molecules also, like metabolites, hormones, yes, neurotransmitters. Uh, so biology is nothing but the activity or interactions between these molecules. For example, when a protein interacts with another protein, that produces a kind of a signal that leads to certain changes. Yeah. So if you want to understand biology, I mean, I think all of you know, people, biology at its core is nothing but chemistry, and chemistry at its core is physics, and physics at its core, I mean, if you want to be a good physicist, you need to be a good mathematician, right? You understand that? So yeah, so I hope this slide is clear to you, because what I'm trying to say is that, see, because when you're doing research in biology or any kind of a thing, I mean, you unless you give it like a mathematical form, or you're able to present it in a using a mathematical kind of analysis, it will all be very kind of fuzzy. I mean, nobody will uh, kind of it's not really valid unless you back it up with certain statistical tests. Yeah, I hope that much is clear. Okay, yeah, just let me know on chat, no, if you're not clear about this, because I think once you're clear about this, then others are much more easier to just go ahead. Yeah. Okay. Okay. Yeah, I will. So I'll show the first slide too at the end of the presentation. Yeah. So again, I mean, see, the whole field of data science or data analytics is trying to do this. I mean, any kind of a data, let's say, yeah, we'll come back to that. And okay, next slide. And now that we know that, see, mathematics is the ultimate form of yeah, you can, what I'm trying to say is that you can describe everything in terms of a mathematical model. Anything, you have biological data, you can describe it as a mathematical model. You have chemical data. So in chemistry, we do mostly like chemical reaction mechanisms and whether a particular reaction is facile. So those kind of things also you can express it in terms of a mathematical equation. Same thing, biology. So suppose if you want to know whether a molecule binds to a protein, again, because see those interactions when a molecule binds to its protein, it uses a series of macromolecular interactions, right? Molecular recognition, these hydrogen bonding, hydrophobic, pi, and each of them has an energy component to it. So when a pi stacking interaction is established, the energy of the overall complex, the small molecule plus the protein, is lowered by 5 kilocalories. Similarly, when a hydrogen bond, so again, I mean, if you can actually present the interaction energies, right, in terms of a mathematical equation. So from there, you know how much energy is lowered, and the amount of energy that is lowered is directly proportional to your binding affinity. Yeah, so we'll come back to that, those things. But generally, I'm telling you, so any kind of biological or chemical or physical, or even if you go much further ahead, no, like this psychological, which are nothing but human, as a human as a whole, and then sociology, which is nothing but interactions between humans, and yeah, so all of them can be modeled using, yeah. And these also, I think, one of the most fundamental slide, because certain softwares, I mean, suppose you're using R or Python, sometimes it can automatically tell you which test to use. Yeah, so again, I mean, see, nominal and ordinal, I think these are the only things maybe new for you, but yeah. So in general, uh, as you all know, when you are applying statistical tests, the first most important aspect to consider is whether your data is normally distributed or not, right? Yeah, so you know normal distribution. Suppose it's like a bell-shaped curve, inverted bell. Yeah, if not, I mean, I just, maybe that's one of the more important. Yeah, I hope you know this thing, right? Normal distribution. So most of the time data is like this. I mean, suppose you're flipping a coin, sorry, let me say how can you tell? Yeah, anything. I mean, let's say, like, you take a set of students in a class. Let's say there are 100 students in a class. You will see most of them scoring between 50 to, or let's say 40 to 60, right? And a very small percentage will score very high, around 90 and above. And again, a very small percentage here will score very low, maybe 10 and below. So mostly they will be roughly centered around the mean will be 50, right? And yeah, so it will range from 40 to 50 or 50 to 70, something like that. I mean, so these kind of a distribution is called normal distribution. And there are tests that are geared towards normal distribution, and generally in research we assume that the data is normally distributed. I mean, that's the because if it is not, we have to make sure that the data has a normal distribution, because I think that's the normal distribution is what we expect in nature, or according to, yeah. uh, because if it is not normally distributed, then something is wrong, then your data either is incomplete or yeah, you understand that. Yeah, so again, I mean, see, basically there are if you look at the type of data, right, there are two types here. The first three are like categorical. So categorical means you don't have exact numbers, but you have just classes. Let's say, like, so, and the other thing is quantitative. Just like, I mean, in the olden days, no, people used to present their mark sheets in terms of numbers, like they got 87%, 97%, like that. But nowadays, maybe you have more of these grading system. So grading is more like categorical, like you have A plus or A or B, or yeah. uh, so when you have these kind of a categorical, which is like your grading system, so then you use these kind of tests like Fisher or Mann-Whitney or student t-test. Again, I think some of you know this thing. I mean, student t-test, most people know. And again, see, there are nominal or ordinal, there are some differences. So both are used for categorical data, and nominal data is that which cannot be ordered. I mean, they're just independent. How can I tell? For example, your grading system, right, can be considered as ordinal because there is a order there. I mean, like, it starts with F, and then it ends at A plus. So there is an order there. I mean, there is a ranking. And nominal is something, they're just independent. I mean, how can I tell you? For instance, like a uh, yeah, like let's say you're in a big city and there are people from multiple states, right? Like let's say from Maharashtra or Karnataka. So again, you cannot just rank them, right? They're all independent. It's not like one is superior to the, yeah. But I hope you understand the difference between nominal and ordinal. So when nominal, we use these series of tests like Fisher and Mann-Whitney, or even chi-square. I mean, that's yeah, for nominal data. And yeah, again, I mean, these are, when you do it, you will understand. I mean, yeah. So these are, but it's good to, it's always this one of the best references that you can keep. And yeah. So again, I hope something is, because you cannot remember everything. That's what I mean to say here. And yeah. Uh, how, yeah, like see, input variable, output variable, is that clear? uh, for instance, see, I'll just give you one example here. Let me for instance, see, you're trying to understand grade, right, as a function of, because suppose you're a teacher, you want to know who will score A plus grade for the next year and who will score F grade for the coming year, right? I mean, so you will just take the data from the year before. So there you will have students from multiple, yeah. So you, but you can model it based on homework time, right? Let's say I call it HT, HWT or whatever, yeah. And I can say breakfast, or yeah, breakfast, and then also like sleep time, right? So using this, so you can see here, grade is nothing but, it is a categorical, but it's ordinal. And these two, right, homework time and breakfast. So breakfast is categorical, but homework time is a continuous, right? Different people have different minutes or hours that they spend. And also sleep also is a continuous. It means, again, it has some, you don't say I slept between 6, I mean, you can say that also, but in general, these two are continuous. So you're trying to establish a relation between a categorical ordinal variable and continuous variables. So yeah, so it will be something like this. So it will be, so continuous is the other term for continuous is called yeah, quantitative. Okay. So again, I mean, if the homework time has a normal distribution, you use something like this, Spearman, or linear. But if it is non-normal, then you use Spearman test. I hope you understand, right? And if it is yeah, yeah, or if you are modeling between categorical and ordinal, and also categorical and ordinal, you use Spearman's rank. Yeah. So I think when you really enter the field, you will know. But these are very, very important, because if you use the test inappropriately, nobody will like to listen to your interpretation. Yeah. And again, uh, in a much more generalized sense, I hope all of you have used calculators and Excel, etc., to make some simple calculations, right? And so those kind of systems like calculators or Excel spreadsheets, they're called expert systems or rule-based models, where the programs are already embedded. They are already inside the, so when you ask 3 plus 2, it will give you 5, because the addition program is already embedded into the calculator or Excel. And so Excel or calculator does this thing. I mean, you give certain data, let's say 3 and 5, and because there is already a program inside, so the computer can tell you, okay, and then you give the function, right? What do you want? You want 3 plus 5, 3 plus 2, and it will give you the output as 5. And but in machine learning, what we try to do is, we have the data. I mean, like, how can I tell you? See, in this case, like I was telling, suppose you want to predict grade using these variables: homework time plus breakfast plus sleep time. So the input data, see the data is homework time here, right? Input data homework time plus breakfast plus sleep time, and the output you want to using these three variables, you want to be able to predict the grade of the student. Yeah, so you can, using machine learning, you can develop a program that can do that. So the program will typically look like this. So the y here is your grade, and so these x, right, x are your uh, input data like homework time. Yeah, so it'll fit it into an equation. So it will say that people who, so the equation typically looks like this, that grade is a function of homework time, and homework time will have a coefficient m. So this coefficient will tell, for example, among the three, right, it will tell if homework time is more important than breakfast or even sleep time. Those kind of, yeah. So so again, I hope there is a small flavor, right, machine learning. And yeah, in terms of machine learning, I mean, machine learning also uses artificial intelligence, because the way, I mean, in a for a simple case, right, we typically have only something like three input variables, or you can call, so this is your dependent and these are called independent. But in a real time scenario, let's say like you're a doctor and you're trying to make a diagnosis. I mean, there is a particular kind of a symptom for a patient, uh, and right, I mean, symptom, for example, linking the symptom with the cause may be sometimes very complex. I mean, the patient may be having a series of illnesses. All of us have many, but they are not so critical that we take care of them. Yeah, I mean illness means anything that is out of the normal. So let's say a doctor, based on various records, right, he has 100 different variables to choose from, and he wants to make a decision, I mean, based on these things like their blood pressure and weight and other things also, like what they're eating for all three times, and where are they sleeping, in what kind of environment, what is the air pollution level, something, all of these. If he considers all of these and he wants to make a decision, so for that he needs to have a model kind of a thing, and that model needs to, you cannot just use all the 100, but typically you use only 3 or 4 in a model. Yeah. So what I'm trying to say is that, see, uh, in when you're modeling, there is an aspect of optimization also. You need to get to the optimal model. Again, how do I tell is that, again, just anybody who knows about these, see, any of you have done this CAD? Yeah, just please show it on CAD, like, or energy minimization. Yeah, any of you know about this energy minimization? Suppose you draw a structure in the computer, you can quickly, because the structure, right, may not have the optimal geometry, both 2D and 3D. Yes. So if you have done energy minimization, right, you know what I'm talking about. Like, the energy minimization tries to get to a structure with some kind of a low minimum energy. And again, people who have done energy minimization also know a technique called molecular dynamics. Uh, uh, so again, molecular dynamics tries, see, energy minimization gets to local minimum, but molecular dynamics, because you are giving it energies, it can search more space, and it has a greater chance of finding the global minima. Yeah. So in terms of modeling also, we use these optimization methods. Yeah, that's because the way, see, suppose you're asked to, let me just go here. Suppose you are asked to, uh, yeah, suppose you have 100 variables, right? Like I was giving the example of the doctor. Suppose you have 100 different, yeah, suppose you have 100 different uh, yeah, just very quickly. see, suppose you, I hope all of you know combinatorial, sorry, permutations and combinations. Because I'll quickly come. I mean, these are some of the important things that are involved in data analysis, permutation and combinations. Yeah, you know this.
Yes.
Yeah. So see here the problem is, see, suppose there is a doctor and he wants to generally learn, okay, when patients have a particular symptom, let's say in this case they have a symptom like impaired what do you call, breathing function or whatever, I mean, yeah, impaired breathing, and he wants to know, can he predict, uh, instead of all the time, putting the patient through various tests and everything, and so can he know just from their vital statistics, and just based on, so let's say patient initially comes and the nurse takes some initial vital stats of him, like blood pressure, everything, and also it gives him a questionnaire to fill up, where is he staying, what is the PMI like, I mean, the air pollution levels, whatever, I mean, so so he can, I mean, let's say he has 100 variables like that, and he wants to be able to get a trend out of it. So he cannot use all 100, right, every time. So he has to make an intelligent model in his mind. Okay, there are 5 parameters which seem to correlate. Yeah. So as you can see, suppose you have given 100 variables and you're asked to select any 5. There are these many different possibilities. So the number is huge, as you can see. Yes. So this is roughly 75 million. That is, let's say, yeah, roughly it looks like 75 crores or something. Yeah. One of the model, right, out of the 75 crore models with 5 variables are used, one of them will be much better than the other. So how do you search for such kind of a model? Is you use these artificial intelligence methods. So there are many like simulated annealing or genetic algorithm, and yeah, there are quite a again this is a huge field, and it's used everywhere in, yeah, in data analysis. Yeah. And finally, also, let's just quickly go through, uh, because as I told you in the beginning, that machine learning makes use of human learning. I mean, it was built on human learning. I mean, they try to mimic how humans learn. And as you know, again, I mean, because you're all students, so you can learn certain things by just trying to memorize them by heart, right? Remembering or memorizing, or you can try to understand also. But when you're asked to apply or evaluate, I mean, these two aspects which are uh, because memorizing and understanding, right, machines are able to do them much better now, because they're meant to do that. I mean, they can store data well, and they can quickly respond to certain commands. Yeah. So memorizing, I mean, any task that involves memorizing, suppose given a face or a fingerprint, suppose you enter a fingerprint, no human will be able to tell or this fingerprint belongs to which human. It cannot tell, right? But a machine can tell, because it has that ability to memorize and understand also, because the whole field of data analytics is trying to derive these kind of an equations. So when you derive this kind of an equation, you get an understanding. I mean, so as I told you, right, when a doctor is confronted with a health record with 100 different clinical attributes, you can call, or clinical data, but he will only look at 4 or 5 when he's making a relation to a particular symptom, right? You understand that? Yeah. So that's because he understood, okay, when a patient has, let's say, impaired kidney function or an impaired liver function, so what he'll do, he'll quickly go to, cholesterol levels could be, so quickly go and check the cholesterol levels out of the hundreds of records, hundreds of entries for that patient, and he'll also look at probably the blood sugar levels, something like that. When he has impaired kidney, he will look at others. I mean, there are these creatine proteins, etc. Because over the time, right, people have developed, or they have developed these kind of an understanding, and machines can do that even better because they are much more rigorous in terms of their trend generation. Yeah, but other things like including creating or problem solving. So this is why, as you know, right, I mean, these all involve uh, like for example, why we still are far from robotics, or autonomous robots, they're called, right? We are still very far. uh, there is some progress, like for example the driverless car. It's a kind of a semi-autonomous. A driver still has to sit in the car, they advise, and yeah. So some of the human learning methods, machines are still far behind, and so they require, for example, like in terms of applying and abstracting or analyzing. Uh, so machines are able to do it, but they need a bit of a human hand, human help. And in terms of judging or evaluating both the same words, and even creating a problem solving, still humans only can are able to do this. Machines are not able to help in these kind of things. But again, so when I say that, there are again examples where there are I'll show you in the coming example, there were very good art or very good music created by just machines, again using some of the methods. Yeah. uh, again, this is also uh, because machine learnings now, I mean, machine learning methods, there is a kind of method called reinforced learning. So as you know, like humans also see, they learn best by doing, they say, right? I mean, rather than receiving or sometimes even participating, just like discussion, like a, yeah, like a back and forth. uh, so machine learning, there are some machine learning methods that are also mimicking this. I mean, these are called reinforced learning, very advanced machine learning. So these are the ones that are able to, or they're able to make music, and yeah, they're doing very, very complicated tasks. They're learning by doing, and that, yeah. Okay, so now we'll not go into all of these because these are yeah. So I just wanted again, you can also, because I didn't cover all of them, right? So again, these are some of the major okay, let me just at least quantitative and yeah. Again, see, the whole data analysis field has uh, three different areas. One is called supervised learning. The other is unsupervised. I think some of you must have already known. For example, clustering is an unsupervised. It means we don't know beforehand how many clusters we would get or something. And some are supervised learning. For example, when you're trying to fit grades, or when you're trying to predict, or you're trying to make a model of grade prediction. So that's supervised. So because you have an endpoint, you know how many grades anybody can get. I mean, you can get from F to A plus, right? So yeah. But in some other cases, you don't know what kind of a class it's called. Like each grade can be called a class. Yeah. And the most recent one are called reinforced learning. And so again, there are many examples. So for supervised learning, there are typically two types: regression and classification. And these are some of the methods that are used: lasso, ridge, knn, spline, xgb. Yeah. And again, supervised learning can also be classification model. And again, unsupervised, we use clustering. And then when you have yeah, factors are nothing but PCA. Yeah, again, these are deep learning, which are nothing but, they're also a kind of a supervised learning, but they go into non-linear kind of models. Yeah, and these are the new ones. Okay. Uh, Again, I mean, so using reinforce, so this deep blue computer, I think it belongs to Google, or yeah, some one of them, or IBM, I think. Yeah. So they even even today they beat the best chess masters. Yeah, like this was, I think, in 1997, Gary Kasparov. So the computer defeated Gary Kasparov. And yeah, like that, there are yeah, it also is playing, I think this also, right, AlphaGo from Google DeepMind. So this is one of the most complex games, they say. It's called Sudoku something. Yeah. uh, so it did well. And then So this is one of the art that recently was generated again by using a machine learning model. So so these are generative adversarial networks. So they can generate. So again, this sold for $500,000. Yes. So, yeah. So like that, there are many. I mean, there are models that can solve these kind of a Rubik cubes. And obviously, we know driverless cars, right? The Tesla driverless car, and even in the legal world, right? Uh, they achieved. I mean, yeah, instead of lawyers arguing and everything, suppose you have various documentation of various evidences. There are softwares now based on machine learning that can tell you who is the real. And they took, as you can see, right, they took far less. They just took 26 seconds, and with more accuracy also. Okay. So one model did the work of almost 20 lawyers, and in a very short time. Yeah. Uh, so it's used everywhere. I mean, as I told you, right, that's why I said, when uh, even judging and evaluating also it is, but then they need some human assistance also. I mean, a human has to look eventually at the decisions that the AI model is making. Yeah. And almost anywhere. I mean, these warehouse logistics, I think this also, right, as soon as certain stock is depleted, it is automatically learning all of this. Yeah. Okay. So I hope, see, till here I covered some of the very basics of machine learning, right? How is everybody? Did I go a little bit overboard? Hi, hello. Yeah. Everybody good? Please tell me.
Good, good. Clear.
Yeah. So in terms of drug discovery, right? I think this slide also some of you must have seen before. See, again, the need is, we have large number of chemicals, at least 20 million compounds that have been synthesized and that are available for purchase, right? There are these zinc databases, there are many other vendors, and then if you look at huge, 10 trillion roughly. And then even drug targets, I mean, protein targets, right? As you know, there are presently roughly around 100,000 proteins, human proteins, and but only 500 are considered as drug targets. And using the genomics, because of genomics, the expected drug targets would increase to 10,000. And so in drug discovery, the challenge is to be able to screen these huge chemical space against the target space. So again, no machine can do that. Whatever amount of resources you have, you cannot do that. I mean, yeah, so only computers can do that. Again, I mean, these are some of the basics. I think somebody who attended my previous talks also would know this thing. I mean, because see, when you're able to model an interaction between a drug and the protein, so you need to so it means that the computer should understand how to represent a chemical molecule and a protein molecule. So it typically represents, I mean, these are the notations, these are the input that we use, miles, or there are other formats also, SDF, mol2. Again, anybody who's done computational work would know. But because in a biological system, right, a protein doesn't care about what kind of a atoms it has or anything. I mean, all that it looks is shape. And then these red and blue, these are called electrostatic potential. So red is negative, and here blue is positive. So if a protein has a pocket, right, that can fit any one of these, it will say welcome. I mean, it doesn't say welcome. So because these guys are moving at randomly at a particular speed, so whenever they see a surface that is complementary to them, like both in terms of shape and then potentials, because this is red, so this will most likely bind to proteins that have a positively charged pockets. And also because see, this size is small, right? So even the pocket should be appropriate to this. I mean, if it is too big, it won't stay. It may come and bind, but it cannot stay. So it has to have a pocket that where this can fit snugly, right? And this has both positive and negative. So it will try to it will try to, it will stay longer in pockets where this kind of a shape and this kind of a electrostatic potential, a complementary electrostatic, like blue should, this blue part will bind to a red part in the protein. Similarly, red will, yeah. So you can model these using computers, right? Because you can estimate the charges. So these are nothing but charges, yes, electrostatic potential, yeah. Uh, so again, I think many of you know this thing. I mean, this is how a charge diagram on a computer looks like. So all that you do is you put a grid. I mean, again, I'll come to this part. Yeah, so you put a grid like this. So grid is a three-dimensional box around the molecule, and you give, you have to mention at what spacing you want certain points placed. I mean, the point is called a probe. So at each of the points or probes, what is the charge that this probe will experience, right? Because it will experience some, see, suppose here you put a O minus, right? And if there is a negative, sorry, if there is a positively charged thing here, it will, it will have, so because it's already negative, right? So it will become even more negative because there is a positive charge here. But you put O minus here, but there is also O minus there. Then it will try to decrease. One of them will have to decrease their charge to, otherwise they will just repel, and that's very unfavorable. Yeah. So what I'm trying to say is that you can measure the charges around the molecule using this grid, because this grid concept, right, is very important. It's used both in QSR, which is like the ligand-based method, as well as in docking. Yeah. So these are very, these are more detailed atomistic details uh, of binding of small molecule and a protein, because drugs are mostly small molecules and their mode of action is against proteins, certain essential proteins. Yeah. In addition to those very detailed models, you can also use some very simpler approaches, because all that we want to know is which drug will bind better to the protein and which drug will have other properties in an optimal way, right? So just like, I mean, how can I tell you? For example, see, again, suppose you're a teacher, right? I mean, maybe sometimes, just by, yeah, for example, the docking and the QSR model, right, that uses the grid concept. So this is like a teacher trying to figure out, okay, because the teacher is going a little more in an advanced kind of a way, trying to understand who will score better. So the teacher, what he can do, okay, he can look at the curriculum for the coming year. And if in that curriculum, let's say calculus is 70% and trigonometry 10, and others, right, geometry, or sorry, others like algebra maybe 10. So based on that, no, and he so now he knows calculus is the most important thing. I mean, anybody who knows calculus well would score better. Calculus and calculus plus trigonometry plus algebra will score even better, something like he has. So he has to go and look at past student records. So anybody, so from past student records, right, he will know that, oh, whenever calculus was heavy in that particular year, a certain student tended to be scoring well. And so so that teacher can map now better, right? Because she knows what kind of a curriculum the coming year has and which student is better able to, he's more comfortable with that curriculum, and so that student tends to do better. And yeah, but other way, I mean, the other kind of model, right, where I showed you, for example, just trying to predict who will do better based on homework time or breakfast, whether they're having breakfast or how much time they're sleeping. So that is like the other kind of QSR. These are called 2D QSR or even 1D QSR, you can call them. And they just look at the properties of the chemical molecule, not of the protein molecule. Again, so because everything, I mean, as you can see, right, one molecule you can describe as a fingerprint. And fingerprint, again, here I just showed few, but there can be 256 or sometimes 4,000 also fingerprints, they call. I mean, so each, so this row, right, if you extend, I mean, it'll go minimum you can have, I think, is 256, just like the bits kind of a thing. and then maximum 4,000, or there are 8,000 and some odd, if you have to multiply the bits. So again, this is huge amount of data, which no human can analyze. Even if you're given 256 columns and 100 rows, right? And for each row corresponds to one molecule with some activity. Let's say it's uh, its its effectiveness as an antiviral molecule, something, yeah. So then you are asked, okay, which parts of the molecule are more important for antiviral? So again, the fingerprint analysis could tell you, because each of them corresponds to one. Like for example, if there is an aromatic ring, you will have a one here, and if there is a methyl group on the aromatic ring, you will have a one. So you can understand. So the second molecule has a methyl, so there is a one there. And similarly, if there is NH2 and O, yeah, so that also is encoded as a fingerprint. Yeah, I hope you're following this, right? So these are some kind of a simplistic models, which is very similar to trying to predict the rank or grade using just the molecule. So just the students, uh, yeah, certain numbers from the students thing only. Yeah, but the other kind of models, docking and 3D QSA, they consider the other side also. For example, the environment also. So when a molecule is binding, so protein is the environment. Similarly, when a student is performing right in class, so the environment is the curriculum. So sometimes when, let's say, calculus is very low, the student who does well in calculus portion is high, so he wouldn't be able to do, right? Let's say in the coming year, I mean, the subsequent year, right after the coming year, in that particular year, let's say the stuff is mostly about by hearting, like memorizing. So in those cases, he won't be able to do, because he has, yeah. I hope you understand. So these are again, see, everything comes to mathematics here. Again, I'll show you how that mathematics is. See, so in drug discovery, these are the two major methods that I use: ligand based and structure based. So structure here means protein structure. So because we don't have protein structures for every protein, that's not there as of now. And 10 years back or 20 years back, the structures of proteins was even to enzyme kind of a proteins for which we had structure, but most of them, but later on, around 15 years back, there were certain advances made, and now we have structures of membrane proteins, all of them. So the structures of proteins has been increasing, but again, there are challenges. Even if you have structure, sometimes you still have to use ligand based, because the structure of the protein, right? I mean, there are multiple functional states, so there will be multiple structural states also of the protein. So sometimes you may not have the structures of the relevant. I hope you understand. For example, see, when you're trying to design an agonist, it means that it will bind and stabilize the most active form. And suppose you want to design an inhibitor, it will bind to the inactive form of the protein and stabilize it. So suppose you don't have structures, right? I mean, sometimes active or inactive structures, they become totally flexible. I mean, because there is no constraint holding them, so they become flexible, and you cannot crystallize certain functional states. So in those cases, you cannot. I mean, suppose you try to design an inhibitor using the active form of a protein, that doesn't make sense, because it means that it will go and bind to the active form, and so you have to use the appropriate functional state of the protein. And yeah, but coming to the mathematics, right? Because we are in the data analytics field. So again, people who are in pharmacy know these things. These are so these are some of the descriptors they're called, or physicochemical properties, right? And then people in pharmacy also know a concept called pharmacophore. And so pharmacophore are nothing but abstraction of various functional groups, right? Like suppose you have an acid, or you have acid is a, yeah, uh, like something like you have a carboxylic acid, and that's an acid. And so acidic group come under negative ionizable, because they can acquire a negative, yeah, uh, so similarly, suppose you have something like an alcoholic group, so that's a functional group. But a broader term would be hydrogen bond acceptor or donor, right? So pharmacophores are nothing but abstractions of various functional groups. And yeah, so see, in QSAR or ligand-based models, we try to so here, just like grade, I said, right? It will researcher would be interested in optimizing his biological activity. Suppose he's working on antiviral, so he wants to have more antiviral effect or antibacterial or whichever, like anti-cancer. So he wants to optimize. So for optimizing, he needs to understand which properties are correlating. If I increase log P, will my efficacy or activity be higher? Or do I need to decrease pKa, or do I need to increase, or by what? So which is more predominant? Suppose given a choice between pKa and MR, right? So MR is molecular refractivity. So which is more important? So if pKa is more important, then he'll try to optimize the acidity or basicity of the molecule. And yeah, yes, I hope. And then see, this is the mathematical model that are typically used in QSARs and pharmacophores. Again, see, there can be many. I mean, anybody who has used QSAR, right, would tell you that there are literally thousands of them. There's a software called Dragon. There are literally 8 or 10,000. So you cannot use. I mean, you cannot when you're developing a model, right, or when you want to present to let's say your managers or somebody that, oh, these are the trends I'm seeing. So the next set of experiments I will design in such a way that I optimize these parameters. I mean, that I will try to design molecules where the pKa is slightly higher than the ones that I made. And similarly, the factors that are detrimental, right, those you want to decrease. So when you design molecule, you make sure that, for example, MR is like the size. Suppose MR is negative to your biological activity. So it means next time you design, you try to design a molecule that has smaller size. And yeah, so all those things. So so in drug discovery, machine learning or data analytics is used in this way. Yeah, and it also gives you a kind of a very, see, that first slide when I told you, right, mathematics is the purest form of thinking. So see, it gives you in a very nice way, where backed up with some numbers also, statistical analysis also, rather than being fuzzy. I mean, you understand what I'm trying to say. So when you analyze data, right, not only has it the ability, you can not only predict better, but you can present your findings better, because you are backing up with statistical analysis. And also you can summarize it much better. I mean, because see, it will, as I told you, right, see, when you have 8,000 descriptors but you want to arrive at a model with 5, you cannot just do that, right? But if you apply a proper machine learning method, it will tell you which is the best set of 5 descriptors that yeah, because it uses these optimization algorithms, and it will tell you which is the best set of 5 descriptors that you can use in the model. Yeah. And similarly, in structure-based drug design, again, some of the biotechnologies people, pharmacy people also might know this thing, that see, there is this Gibbs free energy, delta G. So the delta G, you know, right, change in free energy is nothing but minus RT. This is a constant. Both, yeah, and then minus RT log of Ki. And so from this, you can almost say that Ki, the binding affinity, is nothing but change in free energy, Gibbs free energy, right? This is, yeah. And Gibbs free energy is has 2 components, enthalpy and entropy. And enthalpy component includes these changes in hydrogen bonding acceptor, or yeah, donor, or hydrophobic ionic. And there are many interactions like this. So these can be measured by computer. So when you bring 2 molecules together, right, if they make a hydrogen bond, and there are rules for hydrogen bond, so you know the energy will drop by 2 kilocalories. Similarly, when an ionic bond is made, energy will drop roughly by 4 or 3.5 kilocalories. So you can calculate those, and then once you calculate these, you can, we are not able to calculate the delta ds, is the delta entropy, changes in entropy. These we are not able to calculate that extent. So when you combine both of them, you can predict what is the affinity, right? And this affinity is what's the most important in early stage drug discovery, because initially we try to optimize a molecule against a particular protein, and then we take it to other higher animal models, cell models, all these things. Yeah. Um, so I hope you understand. So again, you see the data analysis is coming here also, even when you're trying to use structure-based design. Yeah. So again, all these are models. So again, see, when you see the kind of interaction energies that are possible, right? There will be many. I mean, because a molecule has, let's say, a molecule like this. So there are, or even a simple molecule, right? See, there can be at least 20 different atoms in a small molecule like aspirin. And if you look at the protein level also, again, there can be 100 atoms in the pocket where aspirin is binding. So you can imagine the combinations, right? And if each atom possibly can make at least 10 different interactions, so the numbers just explode. So, but you want to identify which interaction and which interaction type is the most critical. So again, there also modeling comes in. It will again optimize. It will do that searching, right? Like a global minima, local minima. Yeah. Again, see, this concludes how we are using data analytics in drug discovery. Is everybody clear here? Yeah, because if it is clear, I will go to genomics. Okay. See, if not, you can always ask me. I wanted to show you some success stories also, but because that's not my work. Okay. See, again, how many of you all know genomics? Please, just anybody who knows, you please say yes. Okay. And what percentage you think would know? Yes. Yeah. Any one of you can answer. Almost 70% of you know, right? Okay, that's good. Okay. So again, before we just go into genomics, right? Can you roughly tell me how many genes we have? An average human, right? How many genes are there? Yes, that's good. Okay. And then the idea, right? And you also know these biomarker test. Yes. Like for example, COVID. Yes. And so for example, COVID is trying to measure the quantity, right? It is trying to quantify a certain protein fragment, COVID protein fragment. Yeah. See, I'm not going to epitopes right now. But we can do that. I mean, because see, all of them are very similar. See, just like you're trying to do small molecule, small molecule protein interactions. So again, epitope, they're all very similar. So I mean, in terms of the computational or data analysis, yeah. So again, I mean, I think in genomics, right? The whole idea of genomics is to come up with, again, I'll put it in chat, diagnostic and prog markers. Do all of you know this thing, diagnostic and prognostic markers? So very quickly, diagnostic will tell you disease, this status, yes, and prognostic will tell you whether prognostic is about drug response and survival. Yeah. Yeah. So disease status means, suppose you using biomarker test, right? You want to know, because let's say that cancer happens to be somewhere in a very in some very internal organ, let's say like lung or liver or something, right? You cannot, it's very difficult to do a biopsy and get the tissue, the tumor tissue out of them. Yeah. So in those cases, what we do, we would like to use a non-invasive test, it's called. So one of the best non-invasive tests are genomic profiles of blood, right? You can take blood and and it would have some markers. So diagnostic means it will tell you in what stage it is. So the most advanced stages, they are generally called stage 4 or something, or there are other names also. grade 4, stage 4, and those are very dangerous. It means, but then a doctor, right, unless he gets the biopsy, it's nothing but a tissue of the, a small sample of the tissue, he cannot generally tell. And even that stage 4, it is a very tricky thing. I mean, sometimes somebody can be in advanced, but the tumor has not spread, but somebody is in advanced stage and the tumor has spread. So sometimes that also for a doctor to know could be difficult unless he takes biopsy of neighboring tissues also. Yeah. But a biomarker test. So biomarker is nothing but gene. So you in the test, right, you measure the expression or levels of a particular gene or protein, and that can tell you, okay, based on that, it can tell you whether he's in a very advanced stage and whether metastasis has occurred. And prognostic is where these are the kind of tests, right? Like, for example, certain tumor, I mean, you all know breast cancer, triple negative breast cancer. Yeah, it's also called TNBC. Yeah, people know this thing very quickly. Yeah. So there is a class of no, okay. So there is a type of breast cancer called triple negative. So a triple negative means there are three major proteins, like I think it's called estrogen and progesterone. Yeah. So there are three main receptors for the female hormones, right? Female sex hormones. And these three receptors are generally lacking in those. So they are negative. I mean, it means that they have somehow they're either mutated or changed. And most of the breast cancer drugs actually target those three receptors only, like estrogen receptor, and there are two more. I forgot the names. ERBB2, and there's another one. There are these three. So because most of the drugs target them, and if a patient doesn't have them, it means he won't respond to that drug. So there is no use of putting him on certain certain drug, sorry, certain breast cancer drugs. So because the patient cannot respond to drugs, most likely the patient won't survive for long, right? So even those you can tell by these genomic tests. So it's all about identifying from 25,000 genes. So I call it 25,000. You want to come to a 5 gene test, because you cannot, it's very, sorry, a 5 gene, just 5 gene, 5, or even sometimes people go to even 10 also is okay. But more than that, it will become difficult. Yeah. So you want to for genes which are associated with a disease, right? Yeah. Okay, same thing. Yeah. So again, here you can again see it's about that optimization problem. So suppose 25C5, right? How many numbers would you get? Uh, yeah, it'll also be roughly some number. Yeah, it's something like that. I mean, so most likely you should get somewhere around 53,000 different models, but only some of them will be really good. I mean, to identify the optimal one, the optimal combination of genes, right, that can best explain your breast cancer status. Yeah. So again, I mean, in genomics, right, the data typically looks like this. I mean, I'll very quickly get to show one data set in genomics. Yeah. I'll just open this. See, in genomics, we have to again. I hope you can see my screen, most of you, right? So here you have. So these are all neuroblastoma subjects or patients, and here you have certain clinical attributes. There is an important gene called MICN, whether it's amplified or not, and then you also have age, whether they are less than 18 months, and also here you have staging. Right? You can see, like I told you, staging. Some patients are stage 4, some are 4S, some are 1. The whole idea is that using genomics, right? Because again, see, neuroblastoma happens on the adrenal gland, and adrenal gland, as you know, is next to kidneys or something. Very difficult if you want to do a biopsy, right? And that too for children, much more difficult. So, but is there a non-invasive genomics-based test just by taking a sample of blood? Can you measure the expression of certain important genes and give the staging, right? Because staging is very important, because stage 4 means definitely, I mean, he will immediately, the doctor will immediately do a series of tests and try to remove the tumor surgically if possible, or if it has spread, he has to put him on chemotherapy, something like that. I mean, they are, but then if it is stage 1, then he will go for very mild kind of a treatment methods, maybe little bit of chemo only, not much, and even the monitoring, right, will be very different. He will not use any invasive methods to cut here and there, but mostly using imaging, other things he will yeah. So I hope you understand. See, this is how a clinical file, I mean, we are and then the genomic file looks like this. Let me see if I can yeah, it's a very big file, because if I open this, let's see, if I have already something open, I will quickly uh, something here. So yeah, I'll just one example. See, in this particular data set, which is very similar to the previous one, but here you have only 498 patients. Let me just open one. So this is called R. Yeah, because once you see the data set, I think it should be very easy for you to follow what we do in. Okay, it's opening. or let's open this one. This is what I opened before. See, there are 650. See, these are rows. Okay. Let me just display some part of it. Or even 10, maybe 11 to 20. Okay, so can you see this this particular thing that I'm showing here? Yes, if people can see this, can you please tell me if you can see this? Yeah. Yeah, I hope I hope you can see this. See, what we have here is uh, around 30,000 genes. I mean, because in genomics, right, we measure mRNA expression, and there can be multiple mRNAs, they call, for a particular gene. So sometimes some genes have more than one. So you get, I mean, instead of getting 21,000 or 25,000, you get 30 or sometimes 44,000 also. Yeah. So what it says according to the dimension, right? It says that there are 650. See, this is the dimension of it. See, there are 30,400. Yeah, 30,500 rows. So rows correspond to genes, and 650 columns. Okay. So the columns correspond to patients, and the first column is are nothing but gene names, right? So yeah. So from here, the patient. So these are patient IDs. When we what we saw before, no? So this also had uh, see here also there were 649, right? So you can see six. So it's from the same data set, and for each of the 649 patients, we have certain clinical attributes like what is their which grade they belong to. Now the whole idea is, are there some genes? So each of this is a gene, right? Like hook3 is a gene, or rab24, PAQR8 is a gene. So which of these gene can best explain stage? Okay. So it means that if a clinician can measure the expression or the protein levels of these 5 or 6 genes that we propose, yeah, so he should be able to tell to which stage the patient belongs to, right, without doing an invasive test. Yeah, that's the whole idea. And so now, just very quickly, let's get back to the presentation. And so again, I mean, there are people who can ask different question also, like what makes muscle cell different from skin cell and from liver. Yeah. Again, so again, the technology development is huge, which happened in genomics or life sciences or biotechnology. So I think all biotechnologists would know PCR, right? The standard techniques. So this was I think this was first proposed in 1995. The person got a Nobel Prize. And then PCR even now can be used for one gene at a time. You can measure the levels, right? Expression levels or copy numbers of DNA for one gene at a time. And then during 1995 to 2015, people have been using microarray, which is very similar to qPCR, but or PCR, but it does it on a large scale. I mean, it's like a high throughput. So it, instead of doing one gene at a time, it is doing so the same, the hybridization, the everything. I mean, it follows the PCR protocol, but it does it for many, almost the entire genome wide. I mean, it does it for all the 25,000 genes in a single go, because it uses these things called that microarray chip, which has multiple wells, and each well is a PCR experiment. Yeah. And then since 2015, we have these things called RNA sequencing, which is instead of trying to measure using the hybridization and all these things, right? The intensity that comes from the hybridization, all these things, we are not using anymore. Directly, we are sequencing the RNA or the DNA. And from the sequences only, you can get to know which sequences are higher. So yeah, because so a typical RNA-seq experiment, right? After the RNA-seq experiment, you get this kind of quantification. Yeah, see, yeah, so typically RNA-seq, so because RNA-seq only gives you sequences, right? It will give you short sequences, and yeah, so each of this is a sequence, yes. And suppose this particular thing, this particular region, right, happens to be the genomic location of this particular gene called TMED1. So just by counting the number of sequences, you can quantify. Yeah, that's how. Yeah, but see, in terms of bioinformatics and genomics, currently RNA-seq is used, and to analyze RNA-seq data, for example, as I said, no, you get sequences, and from sequences, you are supposed to quantify the genes, or yeah, you're supposed to quantify genes. So that's a huge undertaking, because you have to align these short sequences to the reference genome, a reference genome, like humans have a particular reference genome, and then after aligning, you have to segment them. Segment means you have to consider these specific areas. Each gene has a specific chromosomal location, right? And within that chromosomal location, you have to count how many are there, and after counting also you have to do some transformations. Yeah, so that's one part of it, and the second part is you again come to this kind of a after you quantify, you again come to this kind of a file. So again, using this, so as I said, no, so this has 649 patients and 30,000 genes, and so for each of the 649 patients, you have something called stage. So stage is nothing but your grade. So some people get grade 1, I mean grade A, some people get grade F. Similarly, in cancer also, some people who have grade 4, right? It means they are in a very bad state. I mean, they are in a very aggressive cancer. Yeah, so again, just like a teacher is trying to predict based on based on the students' homework time and others. So here we are trying to predict the disease state, whether he has cancer or not, and if he has cancer, what stage of cancer, using gene expression. Yeah, yeah. So again, very quickly, let's go back here, and so again, I mean, all these are related to that. Don't worry about these. Yeah, this is how the genomic data sets are presented. So again, see, these are two different cancer types. I think this is glioblastoma, and yeah. So I again, see on the row side, right? Row. This is the row. So you have a set of genes, and you can see that these set of genes, right, are able to distinguish between the two different types of cancer. I mean, they're all one particular cancer only, but one is more aggressive and one is less aggressive. So it means if these genes are high, the clinician can just classify this cancer as GCB like, and because for this subtype of cancer, there are specific drugs that work. Yes, so you understand. So yeah, so all these things, then we use something called survival also. So again, I told you about prognosis, right? Prognosis is nothing but how long the patient will survive. So again, using the gene expression, you can also predict how long the patient will survive and all these things. Yeah, and yeah, these are all. I mean, you will get to know when you come to the field, right? These are all summary. I mean, like, see, when you see in this particular slide, right, in this particular figure, you would have at least 20 genes. But these 20 genes, although they are 20 in number, but they all, let's say, code for only 3 or 4 different mechanisms. Like they're all related to neutrophil, functions like neutrophil migration or whatever. So it will tell you that. I mean, this is just it's called summarization. Yeah, fine. I think, see, even these I try to cover. This is what you get from RNA-seq. And so you can see, right, each of the band is like a sequence. So again, I mean, so see, in this case, this is a normal, and these are patients with some disease like cancer, let's say. So some patients have lower number of a particular exon. Exon, again, you know, right? These are the coding regions within a gene. Gene can have coding and non-coding. Yeah, exonic, intronic, they're called. And sorry, coding, yeah. So again, these also, RNA-seq also can tell you at a exonic level the changes, yeah. Yes. So I think that's about it. I mean, again, there are these things called single cell RNA. So I already told you, right? The data is already quite big for a single human. If you do the whole genome, the typical file size is 100 GB. Sometimes it can be 1 terabyte also, 1 TB also. Yeah, but but in single cell, right? It's much bigger. I mean, it's just because here you're trying to measure for one patient, right? You just don't take one patient as one column. Here you take, for one patient, you measure 10,000 cells. So you can imagine. So suppose you have 649 patients. So the data, it just explodes. So again, a lot of computer science people jump in to help in these kind of cases. And yeah. No, I don't want to bore you, but in general, see, without, I mean, at least in the current life sciences, right, biology, biotechnology, without IT or these data science, data analytic skills, you just can't do anything. I mean, it's not just because even if you join somewhere as a researcher or something, people expect you to identify something that is not already known, right? So you have to develop your own hypothesis. You have to come up with your own markers. You understand what I'm trying to say? For example, see, suppose you're trying to come up with a new painkiller, anti-analgesic, right? So the default target is COX-2. You know, right? Most pharmacy students like because most anti-inflammatory drugs, like all of these, like aspirin and every other all of them, diclofenac, everything targets COX-2, right? But then if you manage to identify a new target, because once COX-2 is in such a stage that there are many drugs, so it's very difficult to identify. Yeah, it's very difficult to identify a new COX-2 inhibitor. And if you don't have new COX-2, new structural chemotype, right? Yeah, it's very difficult to get a it's very difficult to get a patent, a reasonable patent. Yeah, so for that you need to identify a new target also. So if you go back to the pain pathways, there could be another protein as important as so all those things you get from genomics and so I hope you understand, right? I mean, I mean, in genomics, see, finally genomics also can give you drug targets. Somebody has switched on their yeah, fine. Yeah. Please, anybody who is I think all of you try to be in mute because there's no okay. I think hello, who is talking there? Huh? Hello? Yeah. See, I can share it, but I hope somebody from Learn to please. I think you have to make sure the person is muted, please. Yeah, somebody is talking. Yeah, one of them. Okay. Hello. I think it's in. Yeah. Okay. It's fine. Yeah, please. Now, if you have any, if you have any questions, right, you can ask. I mean, because see, these are a little more advanced because although I wanted to cover, but I think you get the gist of it. And see, just like I said, genomics can give you these diagnostic and prognostic markers. It can also give you drug targets. Yeah. Yeah, because see, those markers that I told you right in before, like these things called diagnostic and prognostic markers. So these need not be need not have a driver role, or they need not cause the disease, but after the disease comes, right, their expression or activity can change. Okay, but drug targets have to have a causal role. It means if you if let's say in a disease, yeah, if this particular protein, which is a drug target, if it goes up, that leads to disease. So it means using a drug or a small molecule, if you manage to decrease the activity of that protein, that disease should come down. So you understand. So the drug target has to have a causal role, but diagnostic and prognostic marker, sometimes they do, but that's not a necessary criteria to be a diagnostic or prognostic marker. Yeah. So drug targets, I mean, after you identify certain genes that are correlating with your disease, so you have to, if you establish a causal role and for that gene, and if the gene or protein, right, I mean the protein corresponding to the gene, if it has a proper structure, and that structure has pockets where small molecules can go and fit nicely, so then that can be taken up as a drug target. Yeah. uh, yeah, so genomics gives you all three. It gives you diagnostic and prognostic markers plus drug targets also. And finally, right, see, the whole idea of data science and data analytics is, see, human brain has some again, I told you when we were doing the human and machine learning comparison, right? See, there are certain things that human brain can do well, and there are certain things that machine learning or machines can do well. And for example, analysis of large amount of data, and even I told you, right, this understanding is one of the things that they can do well, because understanding is nothing but trying to summarize also. For example, there are 25,000 genes, and if you ask a machine which set of 5 genes are able to explain, I don't want to, next time when I do the experiment, I don't want to measure all the 25,000, but if you can tell me 5, and using that 5, I should be able to tell the stage or the aggressiveness of a cancer. So machines can do that well. For a human to analyze that amount of data is very difficult. Yeah, yeah, yeah. So so that's the main thing. That's the main advantage of a machine, that it can augment your yeah, your capability. I mean, and you can practically use machine learning, as I told you, right? The applications, they are just exploding. I mean, Israeli researchers, right, or startup companies in Israel, they have come out with apps where, for example, like certain you know, urine tests or something, you know, you had to typically take them and give it to a biochemist, right? But in Israel, they need not do that. They just ask you to collect it in a bottle and take a photograph with your mobile, and just from the photograph that you send from your mobile, they can analyze. They have gone into such levels. And again, there are many new apps now, just using your mobile, right? If you take a picture of your eyes, it can tell you among the 40 eye diseases that are there, which of them you have currently, and with very good. So like that, I mean, the field has exploded, the data analytics field. And yeah, so I open it for questions. Anybody has any questions, please feel free. No, at least do you appreciate, I mean, there are okay, see, the main agenda of this session is to give how data analysis is used in scientific research. So I made it a little more general. I mean, scientific research is not just drug discovery and biotechnology or genomics or life sciences. but I made it general that it is used everywhere. I mean, it's because nowadays there is a lot of interchange. You got Abhijit's side? It's because the title is this, no, data analytics and scientific research. So I just wanted to give an overview. What uh, see in genomics, the other thing is that
Hello.
yeah, yes, I'm here. Hello, who spoke? Yeah, I'm here. I'm listening. Please go ahead.
So in data analytics, jobs and future scope.
Yeah, there is plenty. I mean, it's just plenty. I mean, only thing you should have that ability to pick new methods because the field is changing very, very fast. I mean, for instance, I'll tell you. See, when I was like 2 years back or 3 years back, the GPU technology came. People started, you know, right? Every computation or anything that we do, generally, the processing takes place on CPU, right? Anything visual, like if you're playing video game, that processing takes place on GPUs. Uh, and then what happened is that GPUs became a very hot research area because uh, people started doing scientific computations on GPUs because GPUs, like unlike your CPU, right? CPU you may have at the most 20 processors. Sometimes if you are very capable, 40 also, and in a university kind of a setting, maybe 100 also. But GPUs by default, any small GPU can have several thousand processors, like typically 3, 4,000, 5,000 also. And so that's a huge increase. So whatever used to take a week before, if you have a GPU, proper GPU, you can finish it in 1 hour. So that's the speed up. And now the field has gone into tensors. There are, that's the Google innovation. So accordingly, the methods also change. So the whole idea is if you have sufficient motivation and sufficient basics in the field, and if you are able to quickly adapt to these new technologies, yeah, I mean, opportunities wise, there are just because every startup that is there today is based on these only, these machine learning methods. And yeah, because some of the methods are, some of the startups, right, I know because I used to work in a hospital setting. And so even some of these histopathological images, for example, from a, histopathological images, right, the doctors use, like suppose you give blood or sometimes saliva, they take detailed microscopy images of them. So even to open them before used to take a very long time. Now there are startup companies that are managing to just analyze these in large scale and even display them very fast, unlike before. Yeah, so all of these uh, innovations are happening. And yeah, and machine learning people generally, they I mean, they like to stick to one field, but it's not necessary. I mean, somebody who is good at one can like see, I'll tell you one famous example. The person called David Shaw, you must have heard of him. He was a very successful stock market analyst, but he started as a he was a quantum chemist. You know him, I think. He's the founder of the Schrodinger software and everything. And see, he was initially a quantum chemist, and from there he went into stock market, became very, very rich. And he was Bill Clinton's science advisor, also scientific advisor. And yeah, so I'm telling, so after that tenure with Bill Clinton, he was there 8 years there, and then he came back to academic. I mean, he came back and he founded a software called Schrodinger, which is very successful, widely used everywhere. Yeah. So you understand what I'm trying to say? Computations are like that. You can just uh, because they're all underlying, it's all mathematics and a little bit of physics also. So you can use anywhere. You can use yeah. Yeah, I hope I it's clear. I think he Yeah, he was basically a molecular dynamics guy initially. Yeah. Okay. Next. Now, how many of you are already using softwares like SPSS and Systat? Should I type? Are you using any one of these softwares? Have you used? No? Oh, okay. Uh, no, for your reports, what kind of figures you use? Use Excel only? Suppose you want to put a figure, right, in your thesis or something. Use Excel mostly, or you have not Power BI Excel. Okay, so you, yeah. So again, you see in Excel, you know, some of the things that I told you, right? You can do a t-test in Excel. Yes. Yeah. So same thing. I think you can understand. I mean, but then see, when you do it at a research kind of a level, no, you have to choose an appropriate test. So I told you, right, t-test is used when you have normally distributed data, but when you don't have normally distributed, you go to the other ones, because in genomics we normally use those kind of plots. They're called box plots, and yeah. So again, the box plot has to be backed up with these tests, like, yeah. So if you're a normal student, but if the data is not normal, then you use Man Whitney. Same thing, if the data is normal and if you have more than one category, like instead of just cancer or no cancer, right, you have grades of cancer, like stage 4, stage, because more than 2, you use ANOVA, it's called analysis of variance, and then the data is not normal, Kruskal-Wallis. Yeah. So I think if you have used Excel, you know these tests, some of them, right? Yes. So if somebody knows, please answer. Or has anybody used t-test in Excel? Yeah, go ahead and type, please, if you have used. Yeah. It's fine. You can ask. Yeah. See, I think the most important thing. I mean, if somebody wants to come into data science, right? I think Python and R are highly critical, and there are others also, Scala and all these things. But uh, now Python is the. But in bioinformatics, I mean, if you want to come into the life science sector, and so there R is still the dominant one, almost 95% R is used, but in other areas, other data analysis fields, Python is used, because Python can do, because it is a little closer to C, so it has many more, it's much more versatile. It can do many more things that R struggles to do. Yeah. Yeah, mostly, but then see, you start, see, without having Excel or these softwares I told you, right, Excel or any one of these software, it will be slightly difficult. I mean, it's just that, but then if you know these, it will become much more easier. Yeah, if you know this well, then it's much easier to jump into, uh, yeah, because uh, see, you will get an understanding of the statistical tests, because these are just programming languages. They are not just used for doing statistical analysis, but it can do many other things, right? Python and R, they can do. Yeah. So if you know them initially quite well, like you just try to use the same thing. I mean, you just try to replicate it with Python and R. That's it. Yeah. Yeah. Mostly Python R basics are enough, because nowadays SQL, all these things, right? They're all part and parcel of SQL, or for big data, there's something called Hadoop, uh, and then there's something called Spark. Yeah, yeah, Spark, no, yeah, Spark, yeah, sorry, Spark, Sparkle, yeah, Spark. Yeah, so these two are used for big data. Spark. Okay, let's say. Yeah, so these two, Hadoop, and this is a database query language. There used to be I mean, there are still people, just if you know SQL, right? That SQL, you will generally get a job, and yeah, and there was a time when if you just knew Hadoop, you could get something. But nowadays, because Hadoop and all these things, SQL, they're part of R and Python. So if you know them, you automatically know these also. That's the thing. Yeah. Yeah, and again, the BI, all of these are very similar. I mean, R also has some of the dashboarding. R and Python have the same thing. Boards like Power BI. Yeah. No, undergrad, you may not getting a job directly in US, maybe. But then, see, if you go as a student, it's much easier. But then, if you see the whole thing, acha, you are US, yeah. Then you see, in my opinion, like you should have either a Git, it's called a peer-reviewed paper. Sorry. Or a Git repo with good, yeah. You understand what I mean to say? Like if you display that you have good programming skills and you did some analysis that is peer reviewed and published in a good journal, or sometimes even Git, if you have a considerable Git repository, right, and where people can look up what you have done, then jobs are possible. Yeah, directly. Yeah, because they just want something. I mean, they want because see, in bioinformatics field, as you know, they don't do much of a coding test or anything. I mean, because it's not just about writing code or even doing the analysis, right? You have to write it in a format. I mean, you have to write it up in a as a manuscript. That's the only then, I mean, after you write the manuscript and you communicate and it's accepted, only then your project is completed, unlike in other areas. So other areas, if you just write the software and it is bug free, most likely you need not, your project is done mostly. But yeah, so they're slightly different. I mean, in the software industry, they go through multiple coding tests and multiple interviews, but the bioinformatics job, mostly they look at what you have already the experience of both writing code, doing good analysis, and making a publication. If you can write up a publication, that's yeah. I hope followed, right, Abjit sir. Yeah, but it's easy. I mean, if you know, see, I think if you know Python, just if you know Python, your pay also goes up in US, Canada, some of these countries. I mean, knowing means not just like that you did a course in Udemy or something. You should have actually applied, yeah. Because course is one thing and application. Yeah. Yeah. Yeah, anybody. I mean, I see if you want, I can just show you. If you just open any nature or science paper, right? Almost, I mean, even the citations of people like see all of them. I mean, I'll just show you. See, even the person who was I opened here, his name is Snyder. Almost now, see, every paper that is published or everything, all of it involves data analysis only. So he's a Stanford biologist, but see everything. I mean, if you open any one of his papers, right, you will only see data analysis now. Yeah, because the field is like that. I mean, because now data collection also is very easy, right? From multiple sensors and everything. I mean, everything is miniaturized and almost data is there everywhere. So the only challenge is now to analyze the data. Uh, yeah. So yeah, I hope you got some gist. I mean, I didn't go through much, but yeah, I think so. Yeah. So in this case, I think pandemic preparedness means so maybe you will be using some kind of a, these are called epidemiology. Is the work? Oh, it's related to epitopes. Yeah, yeah. Okay, okay. Uh, okay. Yeah, I think so. See, you will see in a real time how because see some people really ask. I mean, do you really see any advantage of using these models? Because see, in a real scenario, right? Let's say drug discovery also, there are many challenges. Although you have some very good models, but to use the model and come up with a molecule that is better than what you already have is a considerable challenge, because many times the person who developed the model, he wouldn't have enough chemistry knowledge, uh, because when the new design is proposed, right? So if he doesn't consult with the chemist, most likely the chemist won't take it up, because the structure, the chemist will claim it cannot be synthesized, and the chemist will also find faults. Oh, this kind of a structure is not possible, and all these things chemically. I mean, from a synthetic, and and there will be many other things also. Like, even if the chemist likes the structure, right, he may not take it up because he doesn't know the solubility of that molecule. And even for a biologist to assay that particular molecule, he needs to understand to which form of the protein it's binding, because in the assay, right, he has to consider that particular functional state of the protein. I hope you understand all. So there are many challenges. So some people really say, and it requires like a team effort. So I think working in a real time scenario, right? I mean, like somewhere in NTU and on some real data, probably will be quite interesting. Yeah, you will know. Yeah, you will know the reality of it. It's not just yeah, from a theoretical. So epitopes means you will be screening for epitopes, certain. Yeah. Okay. Yeah, it'll be useful. See, anywhere where you, even a simple thing, even if you manage to do something on Excel, and but something statistically rigorous, right? You back it up with some numbers, and yeah, it'll be quite good. Yeah. Has anybody asked? Okay. And next, please. Yeah, please feel free. I mean, this is a very interesting area if you really want to work. Yeah, but it is also very challenging, because that's the whole game. I mean, the software people, they're not paid such big salaries for no reason, right? Because it is a considerable effort, and sometimes just to install a particular package, as we call, or a software, will break you. Like, it's like sometimes it's like that. Right? Yeah, but you have to be really involved. It's nothing like it's something very different from the other jobs, right? That you have to be 100% alert, and your mind should be working. Because when coding, it's like that. You, unless you are like that, you cannot. Please, if anybody has any other question. Yeah, quickly. Also this slide, you could make sense, right? I mean, why? Uh, I mean, why this slide even makes sense? You now understand after you saw everything, right? So because in that particular slide, no, that for the CAD equation slide I showed you, because the whole medicinal chemistry or the whole thing you can represent in these equations, the whole binding event, as we call, no? Yeah. Yeah. So same thing in genomics also. So in genomics, what will happen here? Instead of something like this, here you'll have disease or cancer stage. Okay, cancer stage is a function of certain genes. Okay, like let's say TP53 or MYC, etc. Okay, like so same thing. It means if you can measure these and feed it into the model, it will tell you which stage of cancer you are in. Yeah, I hope you. Yeah. And then some of these, see, these will be called diagnostic or prognostic marker, and if they also have a causal role, then they can be explored as drug targets. uh, so causal role plus they should have structure, and they should have pockets that can be modulated by small molecules. Yeah, the protein should. Yeah, yeah, at least if you follow these two things, right? I mean, in drug discovery and yeah. See, I'm telling you I had to tell you the other things also, right? Well, for example, human learning and machine learning, because it is yeah, because it is also critical. I mean, it's like you need to know where it is currently used and where it is not. Yeah. Okay. Okay. Yeah. So I think I'm done with this. Okay. So if you have any questions, you can get back later, right? Okay. Okay. That's it. Thank you. Bye.
Thank you, sir.
Yeah. Yeah. Bye. Okay. I'm stopping now. Okay. I'm ending the meeting for all, or should I leave the meeting? What should I do?